YouTube and now on Spotify, where we do a deep dive on supplements and longevity studies. Today, we're not just looking at a mechanism of aging. We're looking at what you could almost call the maestro of aging Sirtuin 6, or SIRT6.
That's a perfect way to put it. Sirtuin 6 has been on the radar for a long time as a key player in promoting health span and lifespan. But the real breakthrough we're diving into today is the how. How does it actually do it? What levers is it pulling inside our cells to keep them young?
And it's way more sophisticated than we thought. We're talking about SIRT6 acting like a central control hub for two totally vital internal systems: hydrogen sulfide production to H2S and something called one-carbon metabolism.
Right, which includes a molecule you might have heard of, SAM.
SAM, okay.
And SIRT6's whole mission, really, is to keep your metabolism in a youthful state even as the years go by. See, with aging, these key internal markers, they get all out of whack. H2S production goes down, and SAM levels, they actually go up.
So it's an imbalance.
A total imbalance. And SIRT6 steps in to restore it, pushing those markers back towards their young levels. It's like doing essential maintenance that, amazingly, mimics the effects of caloric restriction.
Okay, let's unpack that. The term Sirtuin gets thrown around all the time in longevity circles. Just to set the stage, what are we talking about here?
We're talking about those NAD plus E-dependent enzymes. Sometimes they're called silent information regulators. They're fundamental. They regulate just about everything: metabolism, DNA repair, you name it. But SIRT6, it's always been a bit of a standout in the family.
Why SIRT6? What makes it so special compared to, say, SIRT1, which gets a lot of attention?
It's the sheer power of the results in overexpression studies. I mean, when scientists genetically engineer mice to have more SIRT6 activity, the effects are just dramatic.
More than just a longer life.
Oh, much more. It's not just lifespan, it's health span. The mice get better glucose control, way fewer malignancies, and they're protected against diet-induced obesity. And the most visible thing is they stay active, like really active, deep into old age. They're essentially delaying that functional decline that we all associate with aging.
That's the dream, right? Not just more years, but healthier, more functional years. So if SIRT6 is doing all this heavy lifting, what are these two internal dials it's turning? Let's start with the molecules themselves.
Okay, so the first one, as you said, is hydrogen sulfide, H2S. It's a gasotransmitter, a bit like nitric oxide, and it's absolutely vital. It's behind a lot of the benefits we get from things like caloric restriction. You need it for cardiovascular function, cognition, tissue repair.
And the problem is, we stop making enough of it as we get older.
Precisely. The research shows that in older mice, the liver's ability to produce H2S drops significantly. So the cells are kind of starved of this critical molecule. That's metric number one. H2S goes down.
Okay, and the second piece of this puzzle, S-adenosylmethionine, or SAM. This sounds a little more chemical.
It is, but it's one of the most important molecules in your body. It's the main methyl donor for almost every methylation reaction.
And methylation is like the cell's post-it note system, right?
Yeah.
Controlling which genes are on or off.
Exactly. If your methylation patterns get messed up, your whole cellular machine starts to break down.
But what happens to SAM when we age? Does it go down too?
And here's the twist. No. Unlike H2S, SAM levels actually increase in the liver of old mice. So aging creates this bizarre challenge. You have less of the good stuff, H2S, and too much of this other essential stuff, SAM. Both are out of their optimal range.
Wow, okay, so this is where the new finding comes in. The big discovery is that boosting SIRT6 in old mice, it fixes both problems at the same time.
It does. It stops H2S from falling and it stops SAM from rising, locking them both into those young-like levels. It's not solving one problem, it's rebalancing an entire ecosystem.
How does it do the first part? How does it boost H2S production?
Well, this is the really clever part. H2S is made through something called the transsulfuration pathway. And SIRT6 uses a surprisingly counterintuitive strategy.
The one you called turning off the delivery track.
Exactly. SIRT6 actively downregulates a transporter on the cell membrane. It's called the XCT system. Its only job is to bring in cysteine, a key ingredient for H2S, from outside the cell.
Wait, so SIRT6 blocks the main supply line for the H2S ingredient? How does that lead to more production? That sounds backward.
That's the genius of it. By reducing the cell's ability to just passively import cysteine, SIRT6 creates a kind of internal pressure, a scarcity. This forces the cell to fire up its own internal production machinery. It starts making cysteine from scratch using another enzyme, and that forced internal synthesis is way more efficient. It massively boosts the overall H2S output.
That's a brilliant feedback loop. It's like shutting down the grocery store delivery so the cell is forced to build its own hyper-efficient farm inside.
Perfect analogy. And what's really important is that SIRT6 does this by acting as a genetic switch. It's not just mopping up chemicals, it's rewriting the operating instructions for the cell.
And what does this boost in H2S mean for the Healthspan benefits we talked about earlier?
It connects directly. This increase in H2S is what leads to those health improvements, specifically a measurable drop in serum IGF-1 levels.
And lower IGF-1 is a classic hallmark of longevity pathways, including caloric restriction.
Yes. So SIRT6 is hitting one of the main longevity dials, achieving a key benefit of CR without having to actually, starve.
That's incredibly powerful. And it really highlights why finding compounds that can activate SIRT6 is such a huge goal in this field.
It is, and that search drives a lot of related research, looking for ways to support this natural function. Sometimes that means looking to nature's own toolkit.
Which leads us perfectly to a really interesting related study. The main paper we're discussing used mice, but there was an older one that looked at natural compounds and their effect on these exact pathways in human cells.
That's right. It was a study on human T cells, and they tested various natural compounds. One in particular, a special form of curcumin called metacurcumin, showed a massive ability to ramp up sirtuin gene expression.
And for SIRT6 specifically, the results were pretty shocking.
They were. The data showed a nearly tenfold increase in SIRT6 expression compared to the control cells, a tenfold increase. And it wasn't just SIRT6, it also boosted SIRT1, SIRT2, and SIRT7. It just shows that certain natural compounds can be powerful allies for our cells' longevity systems. It's exciting to see studies like this discovering how nature's medicine cabinet is vast. That's actually why the team at RevGenetics focuses on unique compounds like physitin, tetracurcumin, and resveratrol since 2007.
Right. The goal is always to support our health using science-backed compounds that can modulate these key pathways, like the one we're seeing with SIRT6's incredible dual role. So let's get to that second half of the job. SIRT6 forces the cell to make more H2S, but you also said H2S can be toxic in high amounts. If it's hitting the accelerator so hard, where's the brake?
That's the other side of the coin, the brilliant countermechanism. SIRT6 seems to understand that too much of a good thing is, well, a bad thing. It has to prevent overproduction. And to do that, it targets a different enzyme called MACHA1. This is the main enzyme that makes SAM.
Okay, so it goes right to the source of the other problem, the high SAM, and slows it down. How?
It uses something called a post-translational modification. Think of MACHA1 as a factory running at full speed. SIRT6 comes in and performs a specific action called deacetylation at a single spot on that enzyme. To use an analogy, it's like SIRT6 puts a governor on the factory's engine. It turns the production line from a high-speed sprint into a more moderate, controlled jog.
So it's not shutting the enzyme off, just changing its behavior, damping it down.
Exactly. This reduces SAM production. And here's the crucial link. High levels of SAM actually supercharge another H2S-producing enzyme called Keeves.
Wait, okay, let me get that straight. High SAM actually leads to more H2S production from a different source.
Correct. So by turning down Meta1, SIRT6 reduces SAM. Less SAM means less activation of Keeves, which puts the brakes on the entire H2S pathway, preventing it from running wild. It's a vital negative feedback loop.
That is just... It's such a dynamic system. SIRT6 isn't just an on switch. It's more like a sophisticated cruise control. It's hitting the gas in one area by forcing internal cysteine production, and at the same time, it's gently tapping the brakes on the whole system by reining in SAM.
That's what makes it such a powerful regulator. It keeps both H2S and SAM right in that sweet spot, that young-like, non-toxic, beneficial range. And this has huge consequences. By managing SAM, SIRT6 also maintains a younger methylation index, the ratio of SAM to another molecule, SAH, and that is absolutely critical for keeping your cells stable and healthy as you age.
So the therapeutic potential here is huge.
Yeah.
We're looking at a pathway that can restore metabolic balance in old age. And since it gives you so many of the same benefits as caloric restriction, the better glucose control, the lower IGF-1 SIRT6 activation could be a really powerful way to get a healthier lifespan without, the constant fasting.
Indeed. And what's really interesting is that the study showed SIRT6's effects were most pronounced in old age or under stress. This tells us its primary role is maintenance and resilience. It's designed to kick in when the stresses of aging really start to build up.
And while this research was on mice, it strongly suggests that maybe combining something like intermittent fasting with targeted SIRT6 activators could create a kind of synergy. You could push that health span envelope even further.
Absolutely. The pathways are all connected. If you can support the same protective systems that fasting triggers, the results could be transformative.
So if we step back for a final thought, SIRT6 isn't just an accelerator for longevity. It's really a sophisticated metabolic homemaker. It's carefully balancing these two critical interconnected pathways to keep the cellular house in a youthful state.
Which leads to a really important, maybe provocative thought for you to take away. Since SIRT6's whole complex mechanism is designed to keep H2S in a very specific beneficial range to actively avoid toxicity, the real challenge for future health science won't just be how do we boost H2S, it's going to be figuring out the precise, safe doses of H2S donors we could take to mimic this perfect internal regulation without crossing that very thin line into causing harm. SIRT6 shows us that balance is everything.
We hope you enjoyed this dive into the content. Thank you for listening to the RevGenetics podcast. RevGenetics.com for the best longevity supplements on Earth.